Hello, everyone. We will wait 1 or 2 minutes for other people to join, and then we can start. Okay, so I guess we can start already. So welcome, everyone. My name is Camelia Markowska. I am project manager for Work Package 6 in Endo-ERN. Today's topic is the new genetics of Turner syndrome. And I do want to remind you that this webinar is accredited by the European Accreditation Council for Continuing medical education. So if you would like to receive a certificate, you just need to fill out the survey that you will receive in your email inbox, and we will send the certificate back to you. I would like to thank MTG7 for organizing this webinar, and also I would like to thank Claus Gravholt for being our speaker today. He is a member of Endo-ERN, and he also works in Aarhus University Hospital in Denmark, and he will be sharing his expertise with you today. All questions are usually answered at the end after the presentation is over. And if you would like to personally ask your question, you can raise your hand, turn your microphone on, and ask wherever you need to. So without further ado, I will give the word to Claus.
Thank you very much. Yes, I'm going to talk on the genomic news concerning Turner syndrome, but also a little bit on Klinefelter syndrome. So you all know Turner syndrome, of course, and there are a lot of different features that we can see in Turner syndrome, but most of these features are really not well explained, except for the short stature, which we now know is due to Schoch's haploinsufficiency. We also know that the median age at diagnosis is about 15 years, and we know that many aspects of body composition, cognition, cardiovascular, fertility, the immune system, and metabolism is affected, and that we have a situation with hypergonadotropic hypergonadism in Turner syndrome. And we know that during adulthood, the treatment is usually estradiol replacement therapy with a progestin as well, while during childhood, we can treat with growth hormone therapy. What I'm going to talk especially about today is body composition and metabolism, but also a little bit on the cardiovasculature. We know that decreased muscle mass and strength is present in Turner syndrome. We know that abdominal adiposity with increased hip and waist circumference is also frequent. And we also know that the occurrence of higher plasma fasting glucose and insulin resistance, as well as the development of type 2 diabetes, the metabolic syndrome, and increased cholesterol and decreased HDL cholesterol are frequent occurrences in Turner syndrome. Then moving on to X chromosome genetics, we know that usually in a normal female, the one X chromosome is almost entirely, or I should say, to a certain degree, inactivated. We've known this for many years, and this leads to the development of the bar body, which can be seen on microscopy. The bar body is really the one X chromosome that is packed tightly together, but there are genes that escape X inactivation here as green dots, as you can see here, and these green dots are expressed also from the inactivated X chromosome. While males have a Y chromosome, but on the Y chromosome, there's actually also genes that are similar to the ones that are on the X chromosome, namely the PAR regions at the tip of both ends of the Y chromosome and the X chromosome. So the PAR genes, the pseudo-autosomal region genes, are also genes that are interesting in Turner syndrome because they are also expressed from both of the X chromosomes in females, but also from the X and the Y chromosome in males. So both the usual escape genes, as we call them, but also the PAR genes are interesting genes, we think, in Turner syndrome. So what do we know by now about the phenotype and the genetics of Turner syndrome? Well, we know that most often we see a 45X karyotype. We know that mosaicism is frequent. We know that there are phenotypic differences between 45X and other karyotypes. And we also know from recent studies from both our labs, but also other labs, that there is a hypomethylation of the DNA in both blood, muscle, and fat, and likely other tissues as well. We know that there are also transcriptomic changes in both blood, muscle, and fat. And then we know from the phenotypic side that getting an autoimmune condition is very, very frequent in Turner syndrome. We also know, of course, that there are frequent occurrence of left-sided heart malformations. And finally, as I said before, the frequent occurrence of metabolic syndrome and type 2 diabetes. However, all these typical phenotypic traits of Turner syndrome have not been linked to any genetic changes in Turner syndrome. So that's what I'm going to touch on during the next minutes here. So as I said, the genes that escape X inactivation may be very interesting. And as I said, the PAR genes are especially interesting, but also the other genes that escape X inactivation, here called XY gene pairs, are interesting genes. I'm not going to touch on all these genes, but I'm going to talk a little bit on some of these genes. We did a study with our American colleagues a couple of years ago, and in this study we found that having one copy of the TIMP1 gene, that is tissue inhibitor of metalloproteinase 1, which is on the X chromosome and usually expressed from both X chromosomes. In other words, an escape gene. So having one copy of the TIMP1 gene, and in addition to that, having a TIMP3 gene normally located on chromosome 22, but with a certain risk allele, was related to having a very high risk of bicuspid aortic valve and lead to aortopathy. And this finding was new, and for the first time, maybe there was a link between some of the genes on the X, and in this case, another gene on one other chromosome, namely chromosome 22. And this finding has recently been replicated by a group from London led by John Ackerman, and they found a similar finding that the combination of this TIMP3 risk allele with the presence of only one type TIMP1 gene was linked to having bicuspid aortic valve or aortopathy. And this finding still awaits functional studies in order to actually show that this is the case. But it's very interesting because then we may finally understand why there's such a large difference between those that have malformations of the left-sided heart and those that do not. So now moving on to neutrophils and inflammation. So this is a study where we actually used several cohorts of patients. We used a genomic cohort with RNA-seq data, DNA methylation data, and clinical traits. And then we did some flow cytometry studies. And we also studied a group of normal females and males together with Klinefelter syndrome patients and Turner syndrome patients. And we also did an epidemiological study and a study of our outpatient cohort. And as you can see here, a lot of different karyotypes were involved in this study. I'll try to take you through the study slowly and discuss with you what we found. So first, this is a plot of on the left side here, a PCA plot of X chromosomal gene expression in 45X patients, the red dots here, and then in 46XX normal females in blue dots. And in the middle here, you can see that all other karyotypes than 45X, but leading to Turner syndrome, are also depicted here. And as you can see, it seems as if when it comes to gene expression, X chromosomal gene expression, the mosaics are in between the normal females and the normal 45X Turners. And then we also discovered that the exist gene, which is the gene that inactivates the X chromosome, which is high on the right here in 46XX females, but low in Turner syndrome because there's no need to inactivate an X chromosome when you only have one. But as you can see here, some of the Turners actually did have a mosaic complement, leading to them having elevated levels of exist close to the normal level in 46XX females, and another group of mosaics that had very low levels of exist comparable to the 45X females. And this exist expression did not, however, seem to be linked to any phenotypic traits. But then we went on to study the methylation data of these patients, and the first plot on the right here, on the left, was of autosomal DNA methylation. And again, you can see that most of the mosaics, green dots here, are in between the normal females and the 45X Turners. However, when we turn to the other figure here, you can see that most, when it comes to DNA methylation of X-linked genes, you can see that almost all the Turners with mosaic karyotypes are very close to the 45X females, telling us that most of the genes on the X are methylated in the same manner as 45X females are. Having done that, we turn to look at the granulocyte proportions estimated using DNA methylation data. And as you can see here, the granulocyte fraction was higher in both 45X Turners, but also in Turners with other karyotypes when comparing them to 46XX females. And when we looked at the neutrophil fraction of the granulocytes, you can see that it also seemed to be higher in both groups of Turners. And this is really not something that has been noted before. So we went on to see if the gene expression levels of specific neutrophil markers were also elevated in the blood from these Turners. And as you can see here, most of these neutrophil markers, these 5 neutrophil markers, you see an elevated level of expression from the leukocytes from the Turners and also from the mosaic Turners. In other words, this could point towards a situation with specific neutrophil markers being elevated in Turner syndrome. We then went on to look at whether we could correlate the gene expression modules with clinical traits and neutrophil levels in Turner syndrome. This is a very busy slide, but just to take out the main results here, we could see that the blue module, which is a module of a lot of genes that were expressed in the same manner in Turner syndrome and in controls, was very closely linked to the neutrophil fraction. There was a strong correlation there. We could also see that the neutrophil fraction was tightly correlated to the heart rate. And this is interesting because, as you can see in panel D there, the heart rate is quite elevated in Turner syndrome. We've known this for many years, and we could also see that in this population that in the controls, the heart rate was about 60, but in Turner syndrome, it was about 70. And this was correlated to the neutrophil fraction, perhaps telling us that the slightly elevated neutrophil fraction was somehow correlated with that. We then went on to look at the neutrophil lymphocyte ratio, and this ratio is another measure of inflammation severity. And when we did that, we could see that many Turners, not all, but many Turners were in the mild to moderate inflammatory level compared with the controls in blue here, where most of them were in the normal range. And panel C here, you can see that we also looked at this in Turner syndrome females compared with normal females and normal males, and in Klinefelter syndrome. And you can see that the more X chromosomes or the more sex chromosomes you have, the lower the neutrophil fraction seems to be. Okay. And then we measured the absolute granulocyte count by flow cytometry, and we repeated our results in an independent cohort here, finding again an elevated level of granulocytes in Turner syndrome compared with normal females. And in this cohort, we could also see that several of the Turners seem to be in the either low inflammatory or the mild to moderate inflammatory range. We could also show that there was no difference in T lymphocytes or B lymphocytes. And in addition, we could see that the granulocyte count was also here correlated to the level of abdominal fat, and that in normal females, there didn't seem to be an increase with age. However, there was a steady increase in Turner syndrome with age in the level of granulocytes. And then we went on to see if we could find any specific genes that were linked with this increased levels of neutrophil. And yes, we were able to see that the gene TBL1X was actually elevated, the expression level was higher in Turner syndrome with only one X chromosome compared with normal females. And we could also see in the population or in the cohort with Turners and Klinefelters and normal females and males that there seemed to be a correlation here in panel B, where the higher the number of sex chromosome, the lower the level of TBL1X expression. And what is TBL1X? Well, it's a gene that is expressed on the X chromosome, and it is expressed ubiquitously, but especially in endocrine tissues, brain and gonads. And it's a downstream target of neutrophil elastase and has been found to be involved in regulating inflammatory cell migration and smooth muscle phenotype modulation in an experimental model of thoracic aortic dissection. And it binds to P65, leading to binding of NF-kappa beta, regulating inflammation and other processes. So we thought it might be a very good candidate gene. So in this part of my talk, I think we believe that we have found a paradigm shifting finding, and we show increased levels of neutrophils and the presence of neutrophil driven chronic inflammatory stress in Turner syndrome. And we seem to be able to link this to an increased expression of the XY homologous gene TBL1X, suggesting a genetic basis for the prevalence of autoimmune disorders and maybe metabolic conditions in Turner syndrome. And we have identified TS individuals with increased neutrophil activation to initiate treatment earlier, which could potentially mitigate the progression towards autoimmunity and more severe metabolic issues. So now I will turn on to something quite different. I'll now talk about epigenetic aging. And the reason for this is, as you all know, we all age. For every year we become older, we age by 1 year, we can say. However, this is when we talk about chronological age. How if we instead talk about biological aging? We all know that we do not age with the same pace. Some seem to be aging faster than others. And therefore, there has been a research area where people have been looking for age-associated biomarkers. And already in 1988, it was suggested that biological parameters of an organism that either alone or in some multivariate composite will, in the absence of disease, better predict functional capability at some later age than will chronological age. So, what are the predictors of biological age, B age? Well, we know that telomere length is something that is important, and we know that the methylome also seems to be important. But also the transcriptome, the proteome, the metabolome, and composite biomarkers play a role. Here, I'll only touch on telomeres and the methylome. And in order to move on with this, people have been investigating epigenetic clocks. DNA methylation is seen as a hallmark of aging due to genome-wide effects of chronological age. Epigenetic clocks are sets of CPGs coupled with a mathematical algorithm to estimate chronological age or age-related traits based on genome-wide studies of large cohorts. And the first generations of these clocks just wanted to see if they could find a DNA methylation algorithm that could explain the chronological age, so the DNA methylation age. And then next generation was interested in seeing if they could predict age-related traits and or diseases and or mortality. And then the third generation of these clocks is also interested in trying to see if they can estimate the rate of aging in individuals. And the Dunedin pace, which I'm going to say something about, is a birth cohort tracking within individual decline in 19 indicators of organ system integrity across 4 time points spanning 2 decades to model the pace of aging. So I'm going to take you through some of the studies that we did on Turner syndrome, but also Klinefelter syndrome. And we also looked at epigenetic age acceleration, EAA. Does that differ from the CH? Is it so that some people age more rapidly and age much faster, while others may have a negative DNA MH, which actually have a de-acceleration of their biological age? And talking about epigenetic age acceleration, there are both so-called intrinsic EAAs and extrinsic EAAs, and this has to do with the way they deal with the cell composition. I'm not going to talk much more about this right now. So how can we use these epigenetic clocks, and why could it be interesting when we talk about Turner syndrome? Well, you may have noted that, or I at least have noted that my patients seem to be aging quite rapidly. And when we look at the loss in lifespan for Klinefelter syndrome, this seems to be about 5 to 6 years when we compare with the normal population. And when we look at Turner syndrome, there is a loss in lifespan of about 13 to 15 years, which is really not explained other than we know that it is primarily due to cardiovascular issues, which can explain the excess deaths, so to speak, in Turner syndrome. So our cohort was a group of normal females compared with Turner syndrome females, and a group of normal males compared with Klinefelter syndrome males. And we then used all these different generations of clocks methylation clocks because we had methylation data on these Turner syndrome and Klinefelter syndrome, and on the normal females and males as well. And so in doing so, we used all these different clocks, and the first results are those that just looked at aging. Could we predict aging? And as you can see on the top here in panel A, where we have Turner syndrome in dark dots and normal females in red dots, you can see that using the data, we predict that Turners are older than the normal females, and we also predict that at least in two of the clocks when we look at the Klinefelter males. So increased chronological age, adjusted DNA age in Turner syndrome, and slightly increased in Klinefelter syndrome. Then we looked at the spread across the different ages of the individuals that were in the study. And for Turners, it seemed that already early on in life, there seemed to be a difference in the slope of the regression line here, indicating that Turners may age more rapidly from early on in life. We then went on to look at telomere length, and in panel A here, you can see that predicted telomere length was shorter in Turner syndrome compared with normal females, again indicating that there may be some kind of early or rapid aging in Turner syndrome. When we looked at the Klinefelter males, it was actually the opposite. The telomere length seemed to be longer in Klinefelter compared with normal males. We did a study many years ago where we looked at a small group of Turners, where we looked at the mean telomere fragment length and found that it was not different from a control group. And another German study here below, where they looked at molecular aging markers in Klinefelter syndrome, finding actually a similar finding as us, that the telomere lengths seem to be actually longer, suggesting reduced biological aging. So moving on to looking at acceleration, we could see that there seemed to be an increased acceleration in Turner syndrome, while this didn't seem to be present in Klinefelter syndrome, at least not to the same extent as in Turner syndrome. So again, moving on to looking at the pace of aging, the Dunedin pace of aging methylation clock is shown here. And as you can see here, the average for the Turners was about 1.1, while the average for the control group in panel A here was actually below 1. So this suggests that for every year a Turner syndrome female grows older, she may age 1.1 year for every year, which again seems to be correlated with already early on in life. As you can see here, it seems that in panel B that there might be an increased aging in Turner syndrome. The results were not statistically significant when we looked at Klinefelter syndrome. So, and this is from the cohort where the Dunedin pace was actually presented earlier on, and here you can see that the similar pattern as we present was present, but in a very large cohort of patients. So what can we use this kind of data for? So we thought that maybe we can use the clinical information that we have in both the Turners and the Klinefelters. And we have performed what we call deep phenotyping. We have characterized both the Turners and the Klinefelters very meticulously, and we now use this data to correlate it to the DNA methylation aging changes that we saw in the different using the different types of clocks here. And this is a correlation plot, and here we correlated the different clocks. You can see the different clocks below there, and we correlated those to the clinical traits that we saw in the Turners and in the Klinefelters. And for instance, we can see here that the correlation matrix was very different amongst the Turners compared with the normal female controls. When we looked at the cDNA, CH, and the DNA age, this was highly correlated amongst females controls with body composition markers such as waist circumference and BMI. However, this was not the case in the Turners. However, when we looked at the Dunedin pace clock, we could see that in Turner syndrome, this was correlated to waist circumference and BMI, and to a certain degree to HDL cholesterol and triglyceride level. In the normal females, the Dunedin pace indicator was not correlated with any body composition measures. However, it was also correlated with triglycerides and total cholesterol. So the correlation matrix seems to be quite different when we look at Turners and normal females. Looking at Klinefelters and male controls, we did not see the same clear, different picture. It was more similar with especially the age and the DNM age and the telomere length correlated to measures of body composition and also total cholesterol. So what we think we can conclude from this is that in Turners versus female controls, there seems to be an increased DNA methylation age. There is also a shorter telomere length, and there seems to be an increased acceleration, both when looking at these different measures of extrinsic and intrinsic epigenetic aging. And the aging pace seems to be increased throughout adulthood, and the aging acceleration seems to be associated with body composition. And we pose ourselves the question, do you grow older faster if your BMI is high, or is it the other way around? We, of course, cannot answer that question right now. Turning to look at the Klinefelters, we could see that there was only a marginally increased DNA methylation age. There was actually longer telomere length in the Klinefelters, and there was only a slightly increased age acceleration in Klinefelter syndrome. And we only conclude that there may be a slightly increased aging pace, and that the aging pace seems to be associated with body composition, both in Klinefelter syndrome, but also in the male controls. So what are the perspectives of this? So if we accept that the methylation profile seems to be increased in Klinefelter syndrome, this may somehow be linked with the increased mortality we see in Klinefelter syndrome, or in Turner syndrome. And as I showed you before, the TBL1X gene seemed to be associated with neutrophil-driven inflammatory stress. And we hypothesized that this may be linked to the immune system, the elevated level of autoimmune conditions in Turner syndrome, but also maybe it is increased, it can explain some of the increased metabolic conditions that we see in Turner syndrome. So these are, of course, hypotheses, and we haven't proved that, but I think this is somehow linking some of the, in this case, one of the XY homologs on the X chromosome to some of these conditions that we see in Turner syndrome. And concerning the cardiovasculature, I have here highlighted that the TIMP1 gene, together with a certain TIMP3 risk allele, seems to explain why some 45X individuals have bicuspid aortic valves and perhaps also aortopathy, while others do not. So it may be that we, with a simple assay, would be able to predict who is actually going to have left-sided heart malformations and who is not going to have that. And then I haven't touched on some other new data, but I'll just show them to you here. And of course, you know that for many, many years we have known that the SHOX haploinsufficiency is associated with height in Turner syndrome. And this was shown many years ago in elegant studies. And this has since been proven by functional studies and other studies. So this is truly something we know is actually taking place. We have also just submitted a paper where we looked at the methylation patterns correlated to neurocognitive traits. And here we found that one interesting gene, the TRAC1 gene, may explain some of the neurocognitive changes, not in Turner syndrome, but in Klinefelter syndrome. And then there are new data on the fertility concerning Klinefelter syndrome with increased intratesticular testosterone concentration linked to microvascular changes and pro-inflammatory responses. And finally, the SLC25A6 gene, which is also an XY homolog, seems to be implicated in QTC interval duration. And this was something we published a year ago. And finally, the ENNP4 gene seemed to be associated with impaired fibrinolysis in Klinefelter syndrome. So where does this take us? Well, I think we may think that the genomics of Turner syndrome and Klinefelter syndrome is becoming untangled, and that subtle changes are present at many sites in the genome, which I hope I have convinced you by showing you the methylation data and the transcriptome data with discrete hypo or hyper DNA methylation affecting gene expression. And that subtle changes in RNA expression are also present, likely leading to unbalanced expression of important genes. I haven't talked a lot about this, but we can see from a study we just did where we looked at the changes in 3 different tissues at the same time. We looked at blood and fat and muscle, and we could see that the changes that we see in Turner syndrome are actually tissue specific. Thus, it seems relevant to examine all relevant tissues in Turner syndrome, and also something like the aorta or the bicuspid valves in order to learn more about what is actually happening there. It also seems that the changes that we see are temporal, meaning that fetal life will likely show very interesting results if we had the chance to look at that. And to that end, there is just a new study out where they actually looked at a few Turner syndrome fetuses and could show important changes there in the ovaries. So I think we now can say that it seems that as if every trait in Turner syndrome has its own explanation, and that we certainly don't understand everything yet, but that we are getting there at an accelerated pace these days. So with that, I will thank a lot of different people that were involved in these studies. They're named here, and collaborators from Portland and from Washington, University of Washington in Seattle, and from other parts of Denmark. And I will also like to acknowledge the funders of the research that we do. And with that, I will be happy to take questions.
So if anyone has a question, you can write it in the question box. It's in the upper right corner. We have not received yet any questions. We can wait a few minutes. Thank you for the great presentation. I think it was very informative, and you showed a lot of graphics, which I believe is also very helpful. So I guess if nobody has questions, we can end the webinar. Thank you for participating. And in the next 2 days, there will be a recording of the webinar uploaded. Yes, Arlene raised her hand, I believe. No? Okay, the recording will be uploaded to our YouTube channel. You can check it right there. And thank you very much, everyone.
Thank
Thank you,
you.
Klaus. And we will see you in the next webinar. Oh, there is a question. Oh, thank you, Arlene. Goodbye.